Welcome to The Explainer. Today, we're diving straight into what has got to be the ultimate medical detective story, a mystery that quite literally haunted humanity for centuries. We're talking about the fascinating history of microbiology. We're going to look at exactly how we went from being these helpless victims of an unseen terror to actually developing the tools, the methods, and, quite frankly, the absolute brilliance required to hunt down our microscopic predators. So, let's jump right in. Picture this. Just imagine the sheer terror of our ancestors. For thousands of years, people observed this one terrifying reality. Diseases spread from person to person. They knew for a fact that if your neighbor fell ill with a plague, well, you might be next. The disease was obviously transmitted by contact, but the actual mechanism? A total void. Without microscopes, they were essentially fighting a completely invisible assassin. How do you defend your entire civilization against an enemy that leaves absolutely no footprints and casts no shadow? It's a question that drove some of the greatest minds in history to start making some remarkably accurate, yet at the time, completely unprovable, guesses. And the initial clues? They actually started appearing way earlier than you might think. As early as the 1st century BC, you had thinkers like Varro and Columella proposing this idea of Animalia minuta, basically tiny, invisible animals causing sickness. Fast forward a bit to 1546, and a scholar named Fracastorius proposes contagium vivum, which is the concept of living germs acting as a separate agent of infectious disease. And then, by 1659, Kircher even reported seeing what he called minute worms in the blood of plague patients. Now, okay, given the equipment of his time, Kircher was probably just looking at regular blood cells, but his intuition? Spot on. These early thinkers made incredibly accurate guesses about living germs causing disease. They just simply lacked the technological equipment to actually prove it. They were basically detectives trying to solve a crime in pitch darkness. So, despite these brilliant early guesses, the entire field of study just lay completely dormant for centuries. Why? Well, because you literally cannot definitively study what is entirely invisible to the naked eye. The science of microbiology was essentially locked behind a massive technological door, and humanity was desperately waiting for someone, somewhere, to invent the key. And that key was the compound microscope. The breakthrough finally happened, and ironically, it didn't come from some prestigious university or a world-famous doctor. No, it came from Antony van Leeuwenhoek, a fabric merchant in Delft, Holland, whose hobby was grinding lenses to inspect the quality of his thread. He started pointing his beautifully crafted lenses at other things, drops of pond water, dental plaque, things like that, and he finally saw them. He saw the microscopic culprits moving around and delightfully named these bacteria little animalcules. Can you even imagine his shock, looking into a tiny drop of water and suddenly seeing an entirely new, hidden universe teeming with life? He actually described the 3 major forms of bacteria and shared his findings, finally proving, once and for all, that this invisible world was real. However, actually seeing the microbes was one thing. Understanding their deadly role and fighting back, that was a whole different ballgame. For a long time, Leeuwenhoek's animalcules were just treated as this weird curiosity of nature. But then, pioneers like Oliver Wendell Holmes in 1840 and Ignaz Semmelweis in 1846 independently realized something huge. We could actually fight back. They noticed that deadly puerperal sepsis, also known as childbed fever, was being transmitted directly by the contaminated hands of medical students and doctors. So they introduced a massive, revolutionary paradigm shift. The solution? Medical hand washing in antiseptic solutions. Just this simple, incredibly basic act of washing hands saved countless lives and proved that we could actually interrupt the transmission of these unseen killers. But to truly go on the offensive, we couldn't just rely on washing our hands, right? We needed to map the enemy's territory. To fight devastating diseases like anthrax, tuberculosis, and cholera, the scientific world desperately needed a rigorous interrogation method. They needed a way to definitively link a specific microbe to a specific crime. And this is where an absolute giant of science steps in to lay down the law in the microscopic Wild West, Robert Koch. Robert Koch established 4 incredibly strict steps, what we now call Koch's postulates. Think of them as the ultimate detective's framework for scientifically convicting a microorganism. First, you have to find the suspect at the scene of every single crime, meaning the organism must be in the diseased lesions. Second, you have to bring the suspect in for questioning by isolating it in a pure culture. Third, the suspect has to recreate the crime. That isolated organism must make a lab animal sick.
And finally, step 4, you have to find the suspect at the new crime scene by re-isolating it from the experimental animal. If a microbe met all these conditions, boom, it was undeniably guilty.
Now, if Koch made us the detectives, Paul Ehrlich transformed us into heavily armed hunters. Ehrlich is rightfully known as the father of chemotherapy, and the sheer genius of his arsenal is just breathtaking. I mean, he developed ways to stain cells and tissues just to understand how they functioned. He reported the acid-fast nature of the deadly tubercular bacillus. He proposed the side chain theory to explain exactly how our bodies produce antibodies. He even introduced methods to standardize toxins and antitoxins. But his absolute crowning achievement was the discovery of specific chemical weapons that we could use directly against disease. And this brings us to a concept that completely changed medicine forever. Ehrlich effectively invented the field of chemotherapy with what he famously called a magic bullet. The idea itself was brilliant, but actually creating it, incredibly difficult. He essentially had to find a specific chemical compound that acted like a microscopic heat-seeking missile. It had to be capable of destroying the disease-causing agent, like the spirochete that causes syphilis, without harming the patient's own healthy tissues. And against all odds, he achieved this with an arsenical compound called salvarsan. For the very first time in human history, we had a targeted drug that hunted the microbe from the inside out, a total game changer. But just when science thought it had cornered the market on microbes, a terrifying plot twist emerged. Scientists had developed these bacterial filters. Think of them as incredibly fine microscopic nets meant to catch literally all bacteria. But researchers, including the great Louis Pasteur while he was studying rabies, started suspecting something else was out there. They took infectious fluids, passed them through these superfine bacterial filters, and obviously expected the resulting liquid to be perfectly safe. It wasn't. The liquid was still deadly. They had discovered agents so unimaginably tiny that they slipped right through the filters. They were ultra-microscopic, too small to be seen even with the very best light microscopes of the day. And just like that, welcome to the terrifying world of viruses. This realization ignited an absolute rapid-fire domino effect of discoveries, basically proving that these submicroscopic assassins were everywhere. In 1892, Ivanovsky proved these tiny agents were destroying tobacco plants. Then in 1898, Loeffler and Frosch found they were responsible for foot and mouth disease in cattle. And then came the really terrifying realization. They hunted us, too. In 1902, Walter Reed discovered that yellow fever was actually a viral human disease transmitted by mosquitoes. And by 1909, Landsteiner and Popper demonstrated that the devastating disease poliomyelitis was caused by a filtrable virus. Plants, animals, humans, literally nothing was safe from this completely invisible threat. The frustration in the scientific community at this point must have been completely off the charts. They knew exactly what the viruses were doing, they could filter them, they could even transmit them to study them, but they still couldn't actually look at them. It took decades of essentially blind research. It wasn't until the invention of the electron microscope by Ruska in 1934 that the veil was finally lifted. Just think about that for a second. 1934. In the grand scope of human history, that is incredibly recent. The electron microscope used beams of electrons instead of light, allowing for vastly higher magnification. Finally, after decades of fighting a literal ghost, we could look these tiny viral killers directly in the eye and study their detailed morphology. But hey, nature always has another secret up its sleeve, right? And this brings us to what is perhaps the ultimate twist: viral oncogenesis. It turns out, viruses don't just cause fevers, or rashes, or paralysis. They can actually trigger malignant cellular transformations. In plain English, they can cause cancer. The clues for this started early. Back in 1908, Ellerman and Bang demonstrated a virus causing leukemia in birds. In 1911, Ross isolated a virus causing foul sarcoma. For decades, researchers desperately wondered if this applied to humans as well. And finally, the definitive positive proof came in 1980 when the human T-cell leukemia virus was isolated. It's a profound and honestly sobering realization. These tiny, invisible invaders can actually hijack our own cellular machinery to cause human malignancy. And that brings us right up to the present edge of our understanding. As we wrap up this explainer, let's just take a step back and look at this incredible journey we've been on. We went from ancient philosophers blindly guessing about tiny animals in the dark, to Leeuwenhoek's little animalcules, to Koch's rigid detective work, Ehrlich's magic bullets, and finally to peering into the incredibly complex, submicroscopic world of viruses. But remember, the human T-cell leukemia virus? That was isolated just a few decades ago, in 1980. Science isn't some finished book sitting on a shelf. It is an ongoing, active investigation. So I'm going to leave you with this final provocative thought.
If it took us this long to uncover the viruses that cause cancer, what other invisible enemies are still waiting to be discovered in the shadows right now? Thanks so much for investigating with me today on The Explainer.